Hey, today we are going to be talking about cells and transport. So, just to kind of show you how this relates to all the stuff we've been doing so far, this picture is showing sort of the levels of organization of living things. So the smallest living thing would be one cell, but we've been looking at things that are smaller than cells. So atoms are the smallest piece of an element, and if you combine them, you can make molecules, including those big macromolecules like our carbohydrates and lipids and proteins and nucleic acids. And then those macromolecules together make organelles, which are almost like tiny organs inside of cells. So just like your body has organs that work together to do functions, your cells each have organelles that work together to do various functions. So the organelles all together make cells. And some kinds of living things only have one cell, like bacteria and archaea, and some kinds of fungi like yeast and protists. But many organisms are multicellular and have many cells, including a lot of plants and animals like humans. And so groups of cells working together make tissues. In Bio 160, we're going to focus mostly on the stuff that cells do. And cells are the basic unit of structure and function in living things, which means that everything that happens in your body, whether it is growth or behavior or movement, all of that has to start with what's going on in individual cells. So that's what we mean by cells are the basic unit of structure and function. But cells can group together, and groups of similar cell types together make tissues. And if you take anatomy and physiology, you're going to talk about the different kinds of tissues in the human body. And then those tissues can group together to make organs and organ systems. So this is a picture of a leaf because leaves are one of the organs of a plant. In humans, we have organs like our stomach and our arteries and veins and heart and brain, all different organs that each have their own function and are made of multiple tissues. The organ systems altogether make the whole organism. So in humans, it's pretty easy to tell what's one organism. You are one organism. But in some species, it's a little bit harder to tell. So if you've ever seen the Himalayan blackberries that grow really commonly around here, you can have a big long row of those blackberry plants that is all one organism, and it turns out they're all connected at the roots, and they will send out these little structures underground that are like a long root that goes to a neighboring area and pops up a new blackberry plant. So some plants, you can, even like some kinds of trees, you can have a whole grove of trees that are all connected at the roots and are all one single organism that has the same DNA. When you have a group of organisms that are all the same species, that's what we call a population. So here we've got one tree in this case. In this kind of tree, one tree is an organism. And a whole group of that species of tree would be a population. We can also talk about, say, the population of humans in Renton, Washington, the population of gray squirrels in Seward Park. When you're talking about multiple different species of organisms, all of the living things in an area would be called a community. So now we're not just talking about the trees, we're talking about all the other plants and the fungi and the animals and the bacteria, and all the living things that live in that area are a community. And then if we add in the non-living things too, like the rocks and the air and the climate and the soil and all of that, that's all part of the ecosystem. So the ecosystem includes the living and non-living things. The community is only the living things. And all the ecosystems together which make up the biosphere, which is the whole Earth. So we've been starting out from small to large. So we started out with the atoms and molecules. Now we're going to move up to the organelles and cells. And then we're going to talk more in this class about, like, how did the cells actually use DNA? What's the DNA do? How do cells signal each other and give each other messages? So some of that kind of stuff too. If you take anatomy and physiology, they'll talk about the tissues, but they're mostly going to focus on the organ systems. So you would go through all the different organ systems in a body. if you are interested in populations and communities and ecosystems, you would probably want to take some ecology classes. Biologists who study populations are often called population ecologists, and then there are community ecologists and ecosystem ecologists. A lot of the people who are studying really big processes like climate change are ecosystem ecologists. But today we're going to focus on the organelles and cells, and how do we get things in and out of cells using transport. So just to review a little bit, we've talked already about prokaryotic and eukaryotic cells. We said that all cells have a membrane around the outside. It's called the plasma membrane. It's made of 2 layers of phospholipids. All cells have DNA, and they are the basic unit of structure and function. Every living thing has at least one cell. And if you want to make new cells, they all come from existing cells. So we can't just make a new cell from scratch. Though there's a field called synthetic biology, and actually a few years ago, a biologist named Craig Venter made a cell that had completely man-made synthetic DNA. So, interesting stuff. You might remember that there are 3 domains that all living things are in. There's the bacteria domain, the archaea domain, and the eukarya domain. So eukarya has all the eukaryotic cells in it. So that's plants and animals and fungi and protists. And then the bacteria and archaea are prokaryotic cells, and they don't have quite as many things. They don't include any of the membrane-bound organelles. And here's this picture that kind of gives you an idea of how much bigger the eukaryotic cells tend to be. So this here would be a eukaryotic cell. You can see there's these organelles that have that sort of little line around the outside, which is the membrane around those organelles. So those kinds of organelles with the membranes around them are only in eukaryotic cells. The purple stuff in there is the DNA. The prokaryotic cell is much, much smaller, and it still has DNA, but it's not inside of a membrane-bound organelle. And it still has a cell membrane around the outside of it, and the eukaryote also has a cell membrane around the outside. We're going to be talking about some of the different kinds of organelles today. And so this is a picture of sort of a generalized animal cell. And we say generalized because there are lots of kinds of animal cells. And even in the same animal, like in the same human, you've got liver cells and muscle cells and neurons and bone cells, all sorts of different cells, and they have different stuff in them and different ratios of different organelles. So not every animal cell is going to look the same. But this is a plant cell, and so some of the big differences you might see between them, and plant cells have different kinds too. There is a cell wall in the plant cell. So this little area right here, this is the cell membrane, and they've cut away part of the cell wall to show you that. The cell wall is on the outside of the cell membrane, and it's thick and protective. And on the animal cell, we've got a cell membrane, but there's no cell wall protecting the outside of it. Cell membranes are pretty thin and flimsy. I think of them like as a really thin plastic grocery bag, like the kind you put vegetables in. And a cell wall is much thicker and stronger and more protective. So like let's say that you were afraid that your groceries were going to break the bag they were in, the plastic bag. So you put it inside of a big cloth canvas bag. That's like the cell wall. So the cell membrane's inside the cell wall. Another difference you might see is that the plant cells have these green organelles called chloroplasts, and animal cells do not have those. So those are involved in photosynthesis, which is the process of converting light energy into chemicals like sugars. So chloroplasts, plants and algae have them, animals do not. The plants also have this thing that looks like a big bubble here called a central vacuole. That's also not found in animals, and it's like a big storage compartment or closet where the plant cells can store extra water and organic molecules like carbs and lipids and stuff. this is a drawing, but in an actual plant cell, I've seen some pictures of them where the central vacuole takes up like 80% of the space in the cell, so they can be huge compared to everything else. So those are some major things that the plants have that the animals don't. So they've got a central vacuole, they've got chloroplasts for photosynthesis, and they've got a cell wall. Really, the only things that don't have cell walls would be animal cells and protists mostly. There's a few other exceptions, but for the most part, everything else is going to have cell walls. Plants have the biggest, thickest cell wall, but like most bacteria and archaea and fungi and stuff, most of them all have cell walls too. So let's take a closer look at the cell membrane. So we talked about with macromolecules that the cell membrane is made of phospholipids. And when people are drawing simple drawings of phospholipids, they will often just draw it as a circle, which would be the hydrophilic head, and then two lines, which would be the hydrophobic tails. Those are made of fatty acids. But they're going to arrange themselves so you've got two layers, and the hydrophilic heads are at the edges, so you got hydrophilic heads over here and over here, and then all the hydrophobic tails are in the middle. So if you think of the lipid, the hydrophobic part, as being like fat or butter or something, it's like a butter sandwich, where the butter's in the middle, that's the hydrophobic tails, and the bread's on the outside, and that's your hydrophilic heads. And that way the hydrophobic tails can stay away from the watery liquid that's on the inside and outside of the cell. You may also notice all these big purple proteins in the membrane. So proteins, we said, have a super complicated shape, and so people tend to get really lazy with their protein drawings, and they're like, I don't want to try to draw that shape, so they'll just make them look like big purple blobs most of the time. Some of those proteins are going to be channels that will allow specific kinds of molecules to go in and out of the cell. Most of the time, the molecules that go through the channels, it's going to only be one kind of molecule that can go through each kind of channel, and they have to be pretty small usually to be able to fit. So anything that's bigger than a monomer probably is too big to go through a protein channel. We also see these proteins here with carbohydrate chains attached to them. Those ones are called glycoproteins. And glycoproteins, they act kind of like identification tags for the immune system. So your immune system, its main job is to go around the body and look for things that don't belong, that are potentially invaders like bacteria or viruses, and get rid of them. And so your immune system really has to be able to identify which cells belong in this body and are part of you, and which cells don't belong in this body and are part of some other organism. And so those carbohydrate chains on the glycoproteins, they are specific shapes, and the shapes of them vary according to your genetics, so you inherit them from your parents. So everybody's got unique shapes of carbohydrate chains on their glycoproteins, and your immune system recognizes which ones are specific to your body. So it'll go around and you'll have these immune cells that have little receptors on them, and their receptors has the shape that just your kind of carbohydrate chain will fit in, and they'll basically latch onto your cell and see if it fits. And if it does, they're like, good, this cell is part of you, it belongs in your body. And if it's the wrong shape and doesn't fit with your immune cell's receptor, it'll be like, this is probably not part of your own body, and it will try to kill that cell. There are plenty of people in medical research trying to figure out ways to genetically alter the receptors on those immune cells so that they will not recognize cancer cells as part of your body and then kill the cancer cells. So we said that there were a bunch of transport proteins, that they tend to be specific. So here are a few ways that people can model or draw transport proteins. So some of them are going to have shapes that can change, like this one. where the transport protein can open on one side and then on the other to allow specific molecules through. Some of them have little gates that can open and close in certain situations. So if you take anatomy and physiology in the future, in your brain, the neurons have a lot of these gated channels where they'll only open at certain times. And then there are also these channels that are open all the time that will allow things to go through. But again, even though the drawing looks like anything could go through, usually it's only one specific kind of small molecule that can go through each channel. So if you want sodium to be able to go through, you got to have one kind of channel for sodium. And if you want potassium to be able to go through, you got to have another kind of channel for potassium. So we're going to look at some different kinds of transport and how things get through the membrane. So we're going to differentiate between passive transport, which doesn't require any energy, from active transport, which does require energy. Usually it's ATP that gives it energy. And so I want to show you this brief animation about diffusion. Let's just turn it into an arrow. Okay, so I'm just going to narrate this. So here we have a beaker of water, and in this beaker of water there's a bunch of molecules that are moving around. So molecules are always moving. Usually the molecules in a liquid are moving faster than the molecules in a solid, but even solid things, the molecules are constantly moving in random directions. And let's say now we want to dissolve some sugar in the water. So here we got a lump of sugar, and even though that is a solid lump of sugar, the molecules in it are still moving around a little bit. So when we drop it into the water, those molecules are going to start to dissolve. So the water molecules are going to get in between them, and as it dissolves, the sugar molecules are going to keep bumping into each other, and that's going to cause them to spread out. So as they bump into each other, they spread out, and ultimately they're going to go from the area where they're most highly concentrated and crowded to the area of lower concentration. And that will continue happening until they're all evenly spread out. So this is called diffusion. So diffusion is when molecules move from their area of higher concentration to their area of lower concentration. It basically means they spread out. People also say that this is moving down their concentration gradient. So when you go from where you're at high concentration to low, you're moving down the gradient. If something were to go from where it's more spread out to being more crowded, we would say that would be moving against your concentration gradient, from where you're at low concentration to where you're at high concentration. So diffusion, things go from their area of high to low concentration. It does not require energy. It will happen automatically. And this happens in air as well as water. So like if somebody on the opposite side of the room is chopping up an onion and it takes a little while and suddenly you smell the onion, it's because the molecules in the onion that are capable of attaching to receptors in your nose so that you can smell them, they got into the air when the person chopped the onion up. And from the air, they started out at high concentration right next to where the person was chopping, and then they gradually moved apart from each other and spread throughout the air by diffusion. Once everything gets to the point where it's at equal concentration everywhere, the molecules keep moving, but we say that they are at equilibrium. So equilibrium, everything's at the same concentration, but it doesn't quit moving. And some things that can make diffusion faster. If you heat something up, that makes the molecules move faster, so that would speed up diffusion. Also, if you have smaller molecules, that's also going to speed up diffusion because bigger molecules, it takes more energy to move them, so they're not going to be as fast. So here we've got another visual. And here we've got these molecules of yellow dye, and they're at high concentration on the left side. it looks like there's a barrier between the left and right sides, but there's some holes in it that the dye molecules can cross through. And so as they bump into each other, they're going to ultimately spread out and go through the barrier to their area where they're at lower concentration. And eventually, at equilibrium, everything will be the same concentration on both sides, but they won't quit moving. Just as many will go from left to right as go back from right to left. At the bottom, we can see what happens when we have two different kinds of molecules. So the yellow dye molecules are going to spread out apart from the other yellow ones, and the purple dye molecules are going to spread out apart from the other purple ones, until yellow and purple are both mixed and there are equal concentrations of both on both sides. So the yellow ones are going to go from their high concentration on the left to their area of lower concentration on the right. The purple ones will go from where they're high on the right to where they're low on the left, and eventually we'll get everything at equilibrium, but it won't quit moving. So we mentioned that this is also called going down your concentration gradient, and that it doesn't require energy, it's passive. Now, water can diffuse too, and there's a fancy word for diffusion of water, and that's osmosis. So here we can see that we've got some solutes dissolved in the water. So we're imagining that the white space is the water, and the green molecules are some kind of molecule dissolved in the water. Maybe it's glucose, maybe it's sodium, we don't know. It's some kind of solute dissolved in the water. So on the left side there are lower concentration of solutes, and on the right side there's a higher concentration of solutes. If there's fewer solutes, that means there's more water. And so water is going to go towards the area that has the most solutes, because then the water is going to try to spread them out from each other, get in between those solutes. So this might seem a little unintuitive, but the solutes, if they could move, the green solutes would go from right to left, because they're higher on the right and they're lower on the left. The water is going to go the opposite direction. It's going to go towards the area with the most solutes. And I'm going to skip this one. Sorry, I must be tired. All right. So this is showing us that there are some terms related to how much water or solutes is in something. And these are usually terms used to compare two areas. Often a cell with the solution the cell is floating in. So let's imagine that we have a cell. Here's our cell. And it's floating in a solution. So the liquid out here is a solution. And I'm going to put some little dots in there to be the solutes. So we're just going to say little red dot is a solute. So I'll put some solutes in the cell. And I'll put a few solutes outside. Those are tiny dots. I hope you can still see them on the video. Okay, so an area that has higher solute concentration is called hypertonic. So in this picture, hopefully you can see that the more solutes are inside the cell. So the cell is hypertonic compared to the solution. An area with lower solutes would be called hypotonic. So the solution has a lower solute concentration. The dots are spread farther apart. So we would say the solution is hypotonic to the cell. So the cell is hypertonic to the solution is hypotonic to the cell. If they both had equal concentrations of solutes, then we would say they were isotonic. So iso means equal, hyper means high, hypo means low. And it turns out that these solutions make a big difference to a cell. So for animal cells that have no cell wall, if you put them in a hypotonic solution, water is always going to go towards the area with the most solutes, so towards the hypertonic area. So if the solution outside is hypotonic, then the cell on the inside is hypertonic. And water always goes towards the hypertonic area. So water is going to go into this cell, and without a cell wall, it could burst or lyse and kill the cell. In a cell that does have a cell wall, like a plant, water's still going to go in towards the hypertonic area, but the cell wall is going to protect it from bursting. So when the water comes in, it will make this plant cell firm or turgid, and so this is actually the normal situation for a plant, is to put it in a hypotonic solution, so the cell is hypertonic, the solution is hypotonic. An isotonic solution, that means both the cell and the solution have the same level of solutes. So in this case, just the same amount of water is going to go in as goes out of the cell. This is the normal situation for an animal cell, like a blood cell. And so if somebody needs fluids really fast, you don't want to give them pure water, because pure water would be hypotonic. It wouldn't have any solutes in it, and it could go into their cells and burst them. So you want to make sure you give the person an isotonic solution, like blood plasma, so that just as much water will go into their cells as out. Now in a plant cell, it's not going to be quite as firm in this situation. It will be flaccid, which means that it like wilts. So a plant will wilt if you put it in an isotonic solution. It's not ideal for a plant. A hypertonic situation is bad for everybody. So if the solution is hypertonic, that means the cell is hypotonic. And water always goes towards the hypertonic area, in this case out of the cell. And that's going to make your cell shriveled or shrunken. So shriveled cells don't work very well. It might kill them eventually. In a plant cell, it shrivels inside the cell wall, so you can see the cell membrane is shrinking. Now there's this extra space between the membrane and the wall. That's called plasmolyzed in plant cells, when the cell membrane shrinks inside the cell wall. One of the ways that many antibiotics work, including penicillin, which is probably our first antibiotic, is that they actually weaken the cell wall of bacteria, certain kinds of bacteria. And when the cell wall is weakened, then if the bacteria is in a hypotonic solution, water can get through and burst the cell wall and kill the cell. So I wanted to real quick show you a practice problem related to this. So there's one in your reading as well. Let me discard this. But let's say that we had a practice problem like, we'll make our font size bigger. Let's say a certain cell, we'll say it's an animal cell, contains 0.2 or 0.3 molar. So this is a unit of concentration. Higher numbers mean more concentration of glucose, and 0.1 molar of sucrose. The solution the cell is floating in has 0.2 molar of glucose and 0.4 molar of sucrose. The cell is permeable to glucose, but not sucrose. And then we'll put the actual questions. So, first, at the beginning, the solution is blank tonic to the cell. And then we'll say Where will glucose move? Where will sucrose move? After the solutes have moved, the cell is blank tonic to the solution. And where will water move? And what will happen to this animal cell? Okay. All right. So we got this animal cell. It's got all this stuff in it, glucose and sucrose. Usually the first thing that I will do in a problem like this is I will draw a picture. So let's draw a simple picture of a cell and a solution. So here's our cell, here's our solution, and we're going to write down what's in it. So the cell has 0.3 molar of glucose. Again, that's a measure of concentration. I'm just going to put G for glucose because I have a hard time writing with a mouse, and 0.1 molar of sucrose. So that's going to add up to 0.4 total. And in the solution, we've got, what happened there? Lost my place, just a second. Alright, I found it. So, in the solution, we had 0.2 molar of glucose and 0.4 molar of sucrose. And so if we add those up, we'll get 0.6. So the lower area, that's our cell, so that's going to be the hypo area. So hypo means lower. And the outer area of the solution has higher solutes total, so that's going to be the hyper area. And so question A asks us, at the beginning, the solution is what tonic to the cell? So the solution, that's the outside, so we said that was hyper. And now it's asking us where things will move. So unless it told us there was ATP, we're going to assume that everything can go from where it's at high concentration to where it's at low concentration. Now also remember that not everything can go through a cell membrane. Only the things the cell is permeable to can go through the cell membrane. So it tells us that the cell is permeable to glucose, so glucose can go through the membrane, but not sucrose. So sucrose cannot move through. So the glucose, it looks like, is 0.3 in the cell and 0.2 outside, so it's higher inside, so it's going to move towards where it's lower on the outside. So glucose is going to move out of the cell. And then sucrose, it said, was not permeable, so it's not going to move. So it doesn't move. And then after the solutes have moved, so once the glucose has moved, it's going to get to the point where it's equal on both sides. So we're not going to pay attention to the glucose anymore. The only difference between the two sides now is the sucrose. So the sucrose is lower on the inside, it's 0.1, and is higher on the outside, it's 0.4. So lower on the inside, it asked us about the cell, that's the inside. So it's going to be lower, so that will be hypotonic. And then water always goes towards the hypertonic area. So if the inside's hypo still and the outside is hyper still, water's going to go out to the hyper area. And this cell was an animal cell, and so if water goes out, it will shrink or shrivel. won't work as well when it's all shriveled. Hopefully you can read that. Anyway, that's how to do those kinds of problems. Let's go back to our PowerPoint. So we're going to look at a few kinds of passive and active transport. So we said that diffusion was a type of passive transport, but there's actually two kinds of diffusion. So there's what's called simple diffusion, which is what's just labeled diffusion on here. So this is simple diffusion. This is when your solute goes straight through the cell membrane, so it can go through the phospholipids. The only things that can do this are small and hydrophobic molecules. So you got to be hydrophobic to get through those hydrophobic tails. So small hydrophobic things can do simple diffusion, which is where they're moving from their area of high to low concentration, and they're going straight through the cell membrane. So some examples of small hydrophobic molecules would be things like O2 or N2, things that have two of the same element, or things that just have a lot of C and H, like CH4. We said CO2 was a nonpolar molecule. Also lipids. So lipids, like your steroids or your fats, those can go straight through. Facilitated diffusion, this is where something goes from its area of high to low concentration, but it goes through a transport protein. And you need a different kind of transport protein for each thing. So maybe this kind of transport protein only moves the triangles, and this kind only moves those blue things. But both are going from their area of high to low concentration. So that's facilitated diffusion. Still does not require energy, it's passive. So this is the way that you get small hydrophilic things through your membrane. So this would be things like ions, so like Na+, K+, Cl-. Ions can go through by facilitated diffusion if you've got a transport protein for that ion. Also, there are facilitated diffusion things for a lot of monomers, so like glucose and certain amino acids. There would be proteins to move those through. All the monomers are hydrophilic, pretty much. There are a few nonpolar amino acids, but most of them is hydrophilic. And then we have active transport. So active transport, the difference is we are going to go from low to high. And it's going to use ATP, so it requires energy. So in this particular case, you can see that this transport protein, this is a protein pump, it's using ATP. This particular one's called the sodium-potassium pump, and it's moving the potassium, which are the diamonds, from where they're low outside to where they're high inside the cell. And then it's moving the sodium, which is the round ones, from where they're low inside to where they're high on the outside. And in order to do that, it requires energy from ATP. So active transport also has two types. This one is called a protein pump. The other kind of active transport is going to be for moving really big things, and that's called bulk transport. So here's another protein pump. It's moving this H+ from where it's at low concentration to where it's at high concentration, and that uses ATP. This is bulk transport, so this is the second type of active transport. And bulk transport is when you move something big, or a lot of little things at the same time. And the way that we do that is that we're going to use a little piece of the membrane called a vesicle. So exocytosis is a type of bulk transport that means you're going to use a vesicle to move something out of the cell. So this little piece of membrane right here, it's got the red molecules inside it, that is a vesicle. And the vesicle can move around in the cell, and it can fuse with the cell membrane, and then basically this is going to split right there. So this is like step 1, step 2, and then step 3. The vesicle's membrane becomes part of the cell's membrane, and now whatever was inside the vesicle is released and can go out. If you're bringing something in instead of pushing something out, then it's called endocytosis. So endo means inside. So here, maybe there's some food molecule or something the cell wants to bring inside. So the cell curves its membrane around it. So this is step 1. Then it curves it around even more. So this is step 2. And then you're going to have a break right here. And so step 3, that piece of the cell membrane is going to turn into a vesicle and surround the thing that we now brought into the cell. So cells can do this to bring in things that they want to eat, like food. They can also do this to bring in big molecules like polysaccharides or proteins that are too big to fit through transport proteins. They can do this to, like immune cells do this, to basically eat bacteria. And I love this animation. So this is a more realistic animation of how vesicles work. The big blue thing on top is a vesicle, and it's attached to this protein that looks like legs. And then there are these long, skinny proteins that look like tracks in the cell. And the motor protein that looks like the legs is actually using ATP to walk along the track and move that vesicle around. And the vesicle is like a big transport truck or something with stuff inside of it that it's carrying around the cell. So, I love that animation. Alright, so now we're going to take a look at some of the organelles in the cell. So first of all, the cell is filled with a fluid that's called the cytosol, or sometimes cytoplasm. You can generally use those interchangeably. There's a minor difference between whether it's called cytosol or cytoplasm. It's like whether it includes the fluid that's in the nucleus or not. And it's a little bit like Jell-O, but generally a bit thinner. It's not quite as thick as Jell-O. Then you have all these structures called organelles that are floating around in that fluid, and a lot of them are anchored to each other and to the outside of the cell with like these long skinny proteins that we call the cytoskeleton. Usually the biggest organelle is the nucleus, so any of these organelles that are big and have membranes around them are only going to be found in eukaryotic cells, so plants, animals, fungi, protists, no bacteria and archaea. So the nucleus is this big round organelle, often one of the only ones you can see under a regular light microscope, and it's full of DNA. So that's where the DNA is. Then you have these transport proteins on the outside of the nucleus that are going to let things go in and out. The nucleus is part of a system of organelles that we call the endomembrane system, and they're all going to be working together to make and modify and transport proteins. So when you want to make a protein, a piece of the DNA, which is called a gene, is basically a recipe for one protein. And in order to keep the DNA safe in the nucleus, we're going to copy that particular gene, which is the recipe for one protein, onto some RNA. And then the RNA copy is going to go out of the nucleus, and when it comes out, it's going to attach to a ribosome, one of these little gold dots here. And the ribosomes, if you zoom way in on them, they look a little like a hamburger bun. So these are tiny organelles with no membranes. These are one of the few that you find in prokaryotic and eukaryotic cells. And they can be free or bound. So this is a photo from an electron microscope. You can see these lines going across horizontally. Those are pieces of membrane on an organelle called the endoplasmic reticulum. And in between them, you have the cytoplasm. And the ribosomes are the little black dots, and you can see some of them are stuck to that ER membrane, and some of them are floating free. So bound ribosomes stick to organelles like the ER, and free ribosomes float around in the cytoplasm. And they can switch back and forth. So if your ribosome is reading some RNA, its job is to read that RNA and make a protein out of it. And so it's reading the RNA, and as it reads the instructions off the A's and U's and G's and C's, it's putting together amino acids to make the protein. And as it puts them together, it may discover this protein belongs in the cytoplasm. And so then it's going to be a free ribosome, and it's going to move and make sure that it's in the cytoplasm. And maybe as it's putting together the protein, it might find out this protein belongs inside an organelle, or it needs to go out of the cell because it's a hormone or something. In that case, the ribosome is going to move over and become a bound ribosome and stick to the rough ER. So this is just showing some ribosomes, actually you have slightly different versions of ribosomes in bacteria versus animals like humans, and you can see that some antibiotics actually disrupt ribosome function by blocking the bacterial ribosomes from being able to make proteins. And without making proteins, your cell's generally going to die because proteins are super important. And so the antibiotics that inhibit bacterial ribosomes are actually not going to inhibit human ribosomes because they're a slightly different shape. So let's look more closely at the endomembrane system and all the organelles that are part of it. We said their job was to make and modify and transport proteins. So this is showing the outside of the nucleus. It's surrounded by a membrane called the nuclear envelope. And then we can see this big blue area, which is called the ER, which stands for endoplasmic reticulum. And parts of the ER are rough because they're covered in these little dots, which are the ribosomes. And then parts of the ER over here are smooth, and they don't have those ribosomes. So they're going to have some different jobs. The smooth ER is mostly making lipids and storing them. So the smooth ER will make lipids like steroid hormones and cholesterol. It will also make new phospholipids so that if your cell needs to make a bigger membrane, it'll make the phospholipids for that. There's special versions of the ER, of the smooth ER, in muscle cells that will store calcium for contraction. And then the smooth ER, also is involved in detoxifying poisons. So like things like liver cells that do a lot of that will have a huge smooth ER. But the rough ER, it's involved in the endomembrane system. So we said that the nucleus had the DNA, and you're going to copy some mRNA onto the DNA. The RNA is going to go out of the nucleus, it's going to attach to one of these ribosomes. And if the ribosome is making a protein that needs to go out of the cell or into an organelle, that ribosome is going to go attach to the rough ER and be bound. When the ribosome builds the protein or the polypeptide, it's going to stick that polypeptide into the rough ER. And then the rough ER can modify it. It's going to fold it up, so polypeptide's a long string of amino acids, but then inside the rough ER it'll fold up so the little partial positives and negatives will stick to each other, and it'll make the right shape of polypeptide. Sometimes it can modify it by like sticking multiple polypeptides together to make a bigger protein, or by adding some carbohydrate chains if it's going to be a glycoprotein eventually. And then a piece of the rough ER membrane is going to break off and become a vesicle, and it's going to have that protein inside of it. So there's this part of the ER over here where pieces of the rough ER can break off and become vesicles that are filled with those proteins that the ribosomes made. And remember, the vesicles walk on the little tracks and they go all around the cell. They're like little vehicles. So then the vesicles from the rough ER are going to go to the Golgi apparatus. And I think the Golgi apparatus looks a lot like a stack of hollow pancakes. And one side of the Golgi is going to receive these vesicles full of proteins from the rough ER. And then it's going to sort them all out and figure out, okay, this one and this one need to go to the same place. We're going to put them together. These ones need to go in the same place. We need to put them together. And then it's going to put them into a new vesicle on the other side of the Golgi apparatus. So it's receiving packages on one side from some vesicles from the rough ER, and then putting them into new packages on the other side. One of the types of vesicles that the Golgi can make, if the vesicle is full of digestive enzymes, it will be called a lysosome. And digestion is breaking things up. So then what the lysosome does is, let's say that this bacteria brought in some food through phagocytosis, which is basically cellular eating. The food is now in this vacuole, which is basically a vesicle that stores stuff and doesn't move around a lot. The lysosome is going to fuse with that food vacuole, and then all these digestive enzymes from the lysosome will break up the food into little pieces so that we can then use it for energy or for using the pieces to build something else. And here's another example where instead of food, the lysosome is breaking down some damaged organelles. So here's a vesicle full of some damaged organelles. The lysosome fuses with it. Its digestive enzyme breaks the digestive enzymes break up the damaged organelles so we can recycle the pieces. We mentioned that the central vacuole was a really large storage compartment found in plants. This is just showing you vacuoles in general are just little storage compartments, sometimes big storage compartments. Vesicles are for movement, vacuoles are for storage. So if we're looking at the endomembrane system as a whole again, it's like an organ system, like your digestive system has a bunch of organs that work together, endomembrane system has a bunch of organelles that work together. So we started out with the nucleus that has the DNA. Some of that DNA, a gene, gets copied onto RNA, and the RNA moves out of the nucleus through a transport protein and attaches to a ribosome. If the instructions on the RNA say to make a protein that is useful in the cytoplasm, the ribosome will be free and it'll float around in the cytoplasm. If the protein we're making needs to go out of the cell or into an organelle, the ribosome will go stick to the rough ER. When the ribosome makes the polypeptide, it will put it into the rough ER where it will be folded and modified by adding it to other polypeptides or to carbohydrate chains. And then the rough ER will put it into a vesicle that breaks off of the rough ER and travels to the Golgi apparatus. The Golgi apparatus receives vesicles from the rough ER and from other places, sorts them all out, figures out where everything needs to go, and packages it into a new vesicle. If that new vesicle happens to be full of digestive enzymes, it would be a lysosome. But otherwise, the new vesicle could go to some other part of the cell, another organelle, or sometimes it does exocytosis. If the proteins in the vesicle belong in the membrane, like they're a transport protein, then this vesicle would fuse with the membrane and the proteins would actually become embedded in the membrane. Or if there's something like hormones that needs to travel to another cell, they would go all the way out by exocytosis. So some organelles that are not in the endomembrane system, here's the mitochondria. The mitochondria are involved in making ATP for energy. And you've probably maybe heard that mitochondria are the powerhouse of the cell, because ATP is basically the currency, like the money that you have to spend to do pretty much anything in the cell. You need ATP. And mitochondria are unique in that they've got two membranes around them, a smooth outer membrane and a folded inner membrane that gives us more surface area for making more ATP. Also, they have their own DNA and special ribosomes, which are a lot like bacterial DNA and ribosomes, and they probably evolved from ancient bacteria that got endocytosed by a eukaryotic cell, but instead of being digested by a lysosome, they started doing useful things. Chloroplasts are the other organelle that is the same way. So chloroplasts also have two membranes and their own DNA and ribosomes, and also probably came from ancient bacteria. These are only in plants. They're what makes the plant green. They're full of a pigment called chlorophyll, and they're involved in doing photosynthesis to help convert light energy into sugars. So this is what I was mentioning, that there's this endosymbiotic theory that a eukaryotic cell long ago endocytosed some kind of bacteria. and then instead of being digested by a lysosome, those bacteria became mitochondria and chloroplasts. So the outer membrane of the mitochondria and chloroplast came from the vesicle, and the inner membrane came from the bacteria. And they actually are full of, their inner membranes have a bunch of proteins in them that are very similar to some kinds of bacterial proteins. So here's some evidence for that. I'm not going to read every single one, but pretty interesting to think that we've got ancient bacteria living in all our cells. So some other things you find in cells, I mentioned that the organelles aren't just floating off in empty space. There's this sort of network of long, skinny proteins that look like nets, and that's the cytoskeleton. So it's going to help the cell to hold its shape, and some pieces of the cytoskeleton are like long, skinny tracks that the vesicles can walk along to get from place to place. Different cells with different shapes, a lot of that is due to the cytoskeleton. So here's one of those cytoskeleton proteins that acts as a track for vesicles to walk along. There are also some proteins that can be on the outside of the cell and help it to move. So some of those are called cilia and flagella. Flagella you may recognize. In humans, the only cells with flagella are sperm cells, and usually it looks like a long swimmer tail that's going to move back and forth to allow the cell to move. Most of the cells that have flagella, they only have one or maybe two or three. They don't have a ton of flagella. So some bacteria can have flagella, obviously sperm cells can have flagella. But cilia, they're also for movement, but they're slightly shorter hairs, and usually a cell with cilia, they will be covering the entire cell, and they'll just beat back and forth. So they don't undulate as much as flagella, but they just move back and forth. So some of your immune cells have cilia all over them to help them move around and like search the body for invaders. Also, the cells in your trachea, in your throat, have cilia. And then if you like inhale a dust particle, the cilia will help push it up so that it can get closer to your throat and you can cough it out. This is a really cool picture. This is a macrophage. It's one of your immune cells, and all the little tiny yellow ones are bacteria. And so the macrophage has these long skinny cilia that it uses to lash out and grab onto bacteria, and then it throws them into its cell membrane so that it can endocytose them, and then lysosomes will digest them inside. That's pretty cool. So here's just a few sort of comparisons between prokaryotic and eukaryotic cells. We said that the organelles with membranes around them, so that includes like your nucleus, your ER, your Golgi, your mitochondria, chloroplasts, all of those are only in eukaryotic cells. Prokaryotic cells don't have them. Prokaryotic cells, they've got a cell membrane, they've got DNA, and they've got ribosomes. They also can have cilia and flagella on the outside, as well as some things called pili that help them stick to each other. And then if we're looking at eukaryotes in general, plants have a few extra things compared to animals. So they've got a thick cell wall made of cellulose, they've got the chloroplasts, and they've got that central vacuole that can hold water and other organic compounds. Animals have the other stuff, but they don't have those ones. One thing to remember, though, is that even within an animal, not all the cells are the same. And so our heart cells and muscle cells and liver cells, they're all different from each other. And they may have different amounts of different organelles. So cells that need a ton of energy and can't ever get a break, like your heart muscle cells, they're going to have way more mitochondria than some other kinds of cells. But all of your cells start out as stem cells. So a stem cell is a type of cell that can turn into any other kind of cell. And it's going to do that by activating and deactivating genes. So this would be like an undifferentiated or unspecialized stem cell. And then it can activate genes that make muscle proteins and turn into a muscle cell. Or it could activate genes that make liver proteins and turn into a liver cell. Or it could activate genes that make neuron proteins and turn into a neuron. The very first cell that makes you, the fertilized egg, which is made of an egg and a sperm, that is a type of pluripotent stem cell, which means it is capable of turning into any kind of cell in the body. There are some kinds of stem cells that can turn into some kinds of cells but not others. So for instance, you have stem cells that could turn into any kind of blood cell, but they can't turn into other things. You might have heard of stem cell research and how it's been, that potentially could help with certain diseases. So cells like your neurons in your brain, they can't regenerate, so if they die, you can't make more. Neurons don't divide to make more neurons. But if you could take a person's stem cells and convince them to turn on the right genes to turn them into neurons, then you could potentially repair that kind of damage in your brain or spinal cord. So that somebody who is paralyzed from a spinal injury, if you could regrow those neurons, you could potentially bring the movement back. So it's one of the reasons people are really interested in stem cell research. So here are just a few specialized kinds of animal cells. So this is muscle cells. They actually have more than one nucleus per cell. Red blood cells don't have any nucleus, and these would be cheek cells. So that little dark purple dot, that's the nucleus in your cheek cells. So different specialized cells are going to look way different from each other and have different amounts of different organelles. And here are some specialized plant cells. So guard cells, these are on the bottom of the leaf, and they have this little hole between them where O2 and CO2 can go in and out of the leaf. And on really hot days, they can swell up and close the hole so that the plant doesn't have water evaporating out of it. Xylem cells, these look kind of like tentacles on an octopus or something, but really they make these long tubes that connect together that are kind of like straws, so that large plants like trees can like suck water all the way up from the roots to the tops of the leaves. And pollen is plant sperm. So if you've ever smelled a plant too close and gotten orange pollen all over your nose, that's plant sperm. You've just been pollinated. Alright, hopefully that helps a little bit with the cell stuff. I posted some extra links with some videos other people have made that are probably better than my videos, honestly, as well as some practice matching and stuff for you.